Hi, I'm Greg Michelotti, and I'm the study director at Metabolon. And today I'd like to provide an overview of how metabolomics provided novel biological insights into sickle cell disease, which is the first genetic disorder for which a causative mutation was identified at the molecular level by Vernon Ingram over half a century ago. Very briefly, sickle cell disease is a devastating inherited hemolytic disorder that affects red blood cell function and is caused by a point mutation that results in the substitution of a valine for glutamic acid at the 6th position of the beta-globin chain of hemoglobin. Now, despite our precise knowledge of the molecular defect associated with sickle hemoglobin in red blood cells, and the recent progress in understanding the molecular events that control polymerization of sickle hemoglobin and sickling of erythrocytes, the mechanistic basis of the disease and associated treatments have remained elusive. In fact, health statistics from a recent World Health Organization report estimated that around 2% of newborns in Nigeria were affected by sickle cell anemia, giving a total of 150,000 affected children born every year in Nigeria alone. In 2015, it resulted in over 114,000 deaths, so despite our knowledge of the genetic mutation for over 60 years, this remains a major global health concern. To generate new insights into the disease, investigators at the University of Texas worked with us to utilize our untargeted metabolomics technology in preclinical models of sickle cell disease, with subsequent findings confirmed in human samples. Results revealed perturbations in several pathways with elevations in circulating levels of adenosine in sickle mice, which is represented by the red dots and by the red arrow on the slide. This being among the strongest signals relative to control mice, which are represented by yellow dots in that z-score plot in the center of the slide. To determine if increased adenosine levels was an effect of the disease or a driver of the disease in vivo, sickle transgenic mice were treated with polyethylene glycol modified adenosine deaminase, or pegylated ADA, which is a drug with a long and successful history of lowering adenosine concentrations in people with adenosine deaminase deficiency. Results show that sickle mice treated with pegylated ADA show marked improvements in circulating hemoglobin levels, which is a typical marker of hemolytic injury in sickle disease. And this is relative to the untreated sickle mice shown in white and highlighted by the red arrow. Now, of course, adenosine is a potent signaling molecule that elicits many physiological and pathological effects via activating G-protein coupled receptors on target cells. So subsequent experiments they identified the direct mechanism of adenosine action shown in this murine model that adenosine-driven sickling is mediated through a PKA, or protein kinase A pathway, downstream of the G-protein coupled receptor adenosine 2P receptor activation. This offers highly targeted therapeutic opportunities using subtype-specific receptor antagonists, which are effective in reducing sickling in both murine and human samples, which are under evaluation currently for treatment options. Now, taken together, these results demonstrate how metabolomics provides unique and actionable insights into a disease for which treatment options have remained extremely limited. More broadly, this approach provides a roadmap for how this technology can be deployed to understand any disease process and identify novel pathways for both treatment and disease stratification.